Hello, Eugene Aiello here. We're going to discuss in this video a little more detail about exactly how electromagnetic energy is absorbed into different pigments, cytochromes, all the things I keep mentioning in this video is about how electromagnetic energy is converted to charge separation. So I figured it was prudent to actually go a little deeper in detail here and understand structures and how this system actually functions, how electrons get transferred into a charge separation by that conversion of electromagnetic energy. So we're going to start first by talking about porphyrin rings. Porphyrin rings are found in heme and chlorophyll. According to most things I read on the internet, they also say that porphyrin rings are found in pigments. Melanin pigment I know does not contain any porphyrin rings, but this might apply to other pigments. I didn't go deep into the research on it, maybe the carotenes, the lycopenes, things like that. But let's go a little detail into how this functions. So first off, a porphyrin ring is composed of 4 pyro molecules, basically carbon structures with a nitrogen on them, double bonds in here, and they're connected together by these bridges, which also are going to contain a double bond. So what this structure creates is a lot of pi electrons. When you're seeing double bonds like this in carbon structures, it means there's electrons which are being loosely held. A porphyrin ring is going to have 26 pi electrons in it, 18 of which are going to be planar. What does that mean? That they're actually oriented in the plane of the molecule. So they're flipping around through here and spinning. They spin so much, those electrons, that they actually create a magnetic field. So when electromagnetic energy comes into it interacts with a porphyrin ring, it's going to excite some of these electrons that are in that outer spin, and it's going to make them more likely to be donated. In, I believe, the next video, we're going to get into that more in detail. These porphyrin rings aren't just sitting there floating within a heme molecule. They are, a heme molecule is composed of a porphyrin ring, but this isn't just floating around in a hemoglobin molecule. These porphyrin rings are connected to protein structures. It could be complex too, with multiple connections. So these electrons aren't just going to be donated to an oxygen. They are going to be donated to an oxygen. They're not just going to be donated to another molecule that's nearby it. They're either, in the case of a heme with an iron center, so hemoglobins, heme molecules, have an iron center that's connected to these nitrogens. That iron center is going to be where the oxygen binds, and that freed electron is going to be donated to oxygen, the cytochromes, as we discussed earlier. Chlorophyll is exactly the same structure of a porphyrin ring, but chlorophyll is going to have a magnesium in the center, which is going to allow it interacting differently. Remember, as we talked about with water, breaking up the water, and with that NADH, with oxygen, that we're converting that electron over for work to be done in the plant cells. So the heme center and a hemoglobin and cytochromes. But what I was getting at earlier is these are connected to other proteins. So hemoglobin is going to have a heme, and it's going to have a protein that's connected to it. So there could be interactions too, as light is absorbed into those electrons within these protein connections. Pigments, those are the other photoabsorbing, electromagnetic absorbing compounds. Pigments are composed of aromatic amino acids. So I think the simplest way to break this down isn't to draw the structure of a melanin molecule and explain how the different pi electrons interact. I think it's best, we're just going to start with explaining how a simple amino acid, an aromatic amino acid, has pi electron structure. So of interest, tyrosine, tryptophan, and phenylalanine are the 3 aromatic amino acids. What that means is an amino acid is composed of basically a simple structure which composes the backbone of all proteins, which in another video coming up, we're going to talk in detail about how these backbones interlink to form protein chains. But the residue, so the difference between every amino acid is what is off of this carbon structure within the backbone. These aromatic amino acids are going to have carbon ring structures with double bonds within them. So there's going to be a pi electron flow within these. I forgot to mention this, I'm sorry. Neurotransmitters, chemical mediators are composed of these amino acids too. I wanted to point that out because later on in videos, this is going to be very important. So these guys are photoabsorbing. So electromagnetic energy is going to excite those electrons. What happens with that is not going to be the same scenarios we talked about with the hemes and with the hemoglobins and the cytochromes and the plant pigments. These guys are exciting electrons for a whole different purpose. We're going to see electron flow, basically. We're creating a charge potential, a separation of charge, which is going to interact with the proteins. And as I stated earlier, I'm going to have a whole topic of the semiconductor nature of proteins. But for now, I just want you to understand that pigments such as melanin are composed of these tyrosines. Melanin, the pigment of our skin, is going to absorb electromagnetic energy from the sun, solar electromagnetic energy, and create a charge separation. Classic conventional biology, we'll talk about the melanin pigments as only being UV absorbers. That is correct. Tyrosine absorbs electromagnetic energy very effectively at the UV spectrum. I think that there's more to that. We're going to discuss in detail later that these different melanin pigments, I keep pointing at it, I don't have a melanin pigment, I'm sorry. Melanin pigments are going to absorb very strongly within the UV spectrum of light. But as you look at the whole visible light spectrum and the near-infrared spectrum, melanin pigments still absorb. They're still pulling in, and if they can absorb it, that means that the energy needed to excite these pi electrons still exists. If a structure absorbs electromagnetic energy and excites an electron, if that excitation state and that energy of the electromagnetic energy that's interacting with it isn't strong enough to create a charge separation, that electron is eventually going to go back down in its orbital. It's going to go back to where it was before that electromagnetic energy excited it. When that occurs, that molecule is going to re-emit that frequency of light. So it won't be a photoabsorber, it will be a transfer. It will absorb it. It wasn't enough to do anything. It wasn't enough to create a charge separation, so it's going to re-emit that electromagnetic energy. So from this video, the most important part of it I wanted you to gather is structures within porphyrin rings and aromatic amino acids absorb electromagnetic energy, exciting electrons for a quantum transfer. I talked about it a little bit in the last video, I believe, that these electrons are getting excited. They're bumping to a higher level, which means they're traveling further from the more attracted area within where the protons and neutrons are within the atoms that are making up that structure. They're getting further away to an excited level. If they never do anything from there, as I mentioned a minute ago, they're going to bounce back to that level and re-emit that energy. With these structures, they're not re-emitting the energy. They're taking that energy in. So an electron transfer is going to be occurring at the quantum level by tunneling. So there's going to be a setup within these structures. In the cytochromes, for instance, it's oxygen. That excitation of that electron is going to bring it to a level that quantum interactions can occur between that oxygen and that electron, so that electron donates over. Within these structures, within a melanin pigment, these structures are going to get excited electrons, but those electrons are going to tunnel to another protein. From there, multiple interactions are going to occur, which we're not discussing yet in this video, but this is a very important concept that as the skin is absorbing that electromagnetic energy, it's not just dissipating it as heat, it's transferring it into a system. And that system is the biophotonic system that I'm going to detail within this series. So thank you for watching this video. I will see you next time.